Hello everybody and welcome to this short lecture on the toxicity of per- and polyfluoroalkyl substances, or short PFAS, within the framework of the program ToxLearn for EU. My name is Henning Hensche. I'm a professor for food safety at the University of Bonn in Germany. And I'm active in scientific research and in academic teaching in the areas of food safety and food toxicology. So that fits very well to the topic of today, toxicity of PFAS. The presentation is structured in 4 chapters, starting with hazard identification, second hazard characterization, exposure assessment, and risk characterization. So the presentation follows the general structure of the classical risk assessment. So we start with the hazard identification and looking at the PFAS, what type of compounds they are, what different substances there are. So it's a chemical identification. Then in hazard characterization, we're talking about the toxicology of the PFAS. So we're looking at the toxicokinetics, we're looking at the dynamics, so which mechanisms are involved, and then also which effects occur and which health-based guidance values have been established. Then in the third step, we look at the exposure, so how much PFAS do we actually take up, mainly via food. And then in the last step, in the risk characterization step, we bring all of this together. And we start with the first chapter, the hazard identification. PFAS, per- and polyfluoroalkyl substances, as the name already says, are alkyl substances where all carbon-hydrogen bonds are substituted by carbon-fluorine bonds. You can see that here in the general structure. So we do have alkyl substances that also have functional groups, particularly carboxy group or sulfoxy groups, and then we don't have hydrogen atoms here, so there's no carbon-hydrogen bonds, but all hydrogens are substituted by fluorine atoms. And this also gives the special characteristics of PFAS, because the carbon-fluorine bond is very, very stable. And that means that PFAS are also very stable compounds as such. So they're very stable against high temperature, against chemical insults, and this is the most important property of the substances, why they are used in various industry applications. One of the very typical substances you can see here, P4S, perfluorooctane sulfonic acid. So again, here you see, as in the general structure above, you see the alkyl moiety, octanoic acid, but all the hydrogen carbons are replaced by fluorine carbons. And then we have the sulfoxy group here. Now, P4S is one of the major compounds and one of the most important ones, certainly. But PFAS contain a huge amount of different substances. You can see here a small selection of those compounds that where frequent industry applications have been identified, but there's much more to it. Estimations go into the thousands, so approximately so thousands of compounds are chemically known. Of course, not all of them are used technologically, and not all of them are relevant for environment or human toxicology. But nevertheless, it's really a huge compound class, and many compounds are considered toxicologically relevant, even those where we don't really have a lot of toxicological data. On this side, you see the different uses of PFAS. So you see on the left-hand side different industry sectors, and on the right-hand side different products or items that contain PFAS or that use PFAS. You see the total amount of substances used. We don't need to really look at the numbers. This is just to get an impression of how many different industry sectors and products are actually using PFAS. So you see, for example, here in the industry sectors, electronic industry has a high amount of PFAS being used, and we have production of plastics and rubbers that has really a huge amount of polymers. And looking at the At the products, we have coatings and paints, and this is particularly the coatings because, as I said, PFAS are highly resistant, and especially to chemical degradation and also to high temperature. So this is why these PFAS are being used in products that need to be resistant to these influencing factors. Lubricants and greases are also being used, and you see that quite a number of different products do contain or did contain PFAS. Now, the use overall is declining of PFAS, mainly due to the fact that people recognize the toxicological problem associated with the PFAS. Some of you might have heard of the term forever chemicals that is used to describe PFAS, mainly because they have such a long half-life in the environment. So once substances are out in the environment, either by leaking from these industry sectors or by the different products being used by the consumer, they are very, very stable and they're not degraded. Unlike other compounds that are being degraded, say at least within months or years, these substances, the PFAS substances, are usually stable for many decades and maybe even longer in the environmental ecosystems. And that already brings me to the first breakout question. Now, at the end of all the different chapters, of all the 4 chapters, we will always have a breakout question where you can yourself answer one single question or maybe also further questions. I would like to ask you here to check on the internet or maybe other documents that you have available how the use of PFAS is regulated. And this is mainly with the background of what I just said before, that there's growing awareness that PFAS are a major ecotoxicological and also human toxicological problem due to their very long half-life. And the question is, how are PFAS's use being, how is the use being regulated? Maybe also for those of you who are already aware of the fact how the group of PFAS is regulated or planned to be regulated, you can also look at individual substances and see which substances are maybe banned already, where are restriction limits, high maximum amount limits in food, etc. Okay, and with that we come to the second part, the hazard characterization. So here we look at the toxicological properties of the PFAS. We start with the toxicokinetics. PFAS are usually absorbed rapidly and also completely. Interestingly, there's no relevant metabolism, which is usually not the case for toxicologically relevant compounds. There's usually some kind of either toxification or detoxification, but we don't have that with the PFAS because they are so stable and basically inert substances that we've seen already in the hazard identification chapter. This is also true here in the metabolism. So they are taken up in the body, but they're not being degraded or changed chemically or metabolically. So there's no metabolism. The substances are excreted to a slight extent, but there's very strong reabsorption mechanisms. So we do have enterohepatic circulation. So though the fraction of the substance that is being eliminated hepatically is being taken up into the body again, into the central compartment, into the blood, and also the part that is being absorbed by, that is being eliminated, by the kidney is being reabsorbed within the renal systems. And this is why there's very long half-lives. The compounds as such are very stable. There's no metabolism, so they're not altered chemically. And they're also eliminated to a very, very slow extent. And this is why they have very long half-lives. What I mentioned already before in the ecosystems is also true in the human body. And what you see on this slide is the half-life in numbers. So what you see here is data from one of the latest BFR opinions. The BFR is the German Federal Institute of Risk Assessment. The BFR frequently publishes opinions on various topics of toxicological interest, but actually, in fact, the data presented here are taken by BFR from the EFSA opinion from 2020, which I will mention later on. So what you see here is the numbers of half-lives of different PFAS compounds in different species. You see here the various substances. They are first of all stratified into sulfonic acids and carboxylic acids, and then they are ordered by their chain lengths. The chain length of the compounds is a very important parameter determining the half-lives, as you can see here. So we start from very short chain lengths, PFBS, up to PFOS, medium or large chain lengths, and then for the carboxylic acids as well, starting from the very short compounds, short chain compounds, up to very long chain compounds. And you then see here different species, various experimental models, animal models, and also humans. And what you can clearly see is, first of all, when you only look at humans, that there is a clear dependency of the half-life depending on the chain length. So very short-chained compounds have half-lives in the range of days, as for PFBS here, and also PFBA, PFHXA. But then when we go higher, perfluorohexane sulfonic acid, PFOS, then we're in the area of years already, and that can be up to 7 or 8 years in half-life. So this is really, these are very strong, very long half-lives in comparison to other toxicologically relevant compounds. Typically, when we don't have any accumulation, we have half-lives in the area of days or weeks, but here we are at least in the area of many months, maybe even, or for some even years. What you can also see, interestingly, is that half-lives are quite different in the different species. Now, when we only look maybe at PFOA as one typical compound, you see that for humans we do have this very long half-lives of years, but especially the rodents have very short half-lives, and also monkeys or pigs, which are considered kinetically more similar to humans, of course, have half-lives in the range of days, not months or years. So there's also a clear species-specific effect. Looking at the adverse effects, so the question which effects are being caused by PFAS, you can see that here in the picture many different organs, organ systems or tissue types can be affected. PFAS really can have a vast variety of different effects. The most important ones are named here: hepatotoxicity, so liver damage; immunotoxicity, so damage to the immune system; reproductive toxicity, so fertility, for example; and also cancer at high doses. In the previous EFSA opinions, hepatotoxicity and immunotoxicity were the critical effects. The health-based guidance values that I will mention later on were in 2018 defined based on studies on hepatotoxicity, and now the last opinion from 2020 from the EFSA used immunotoxicity as critical endpoint. I will mention that later on again. Regarding the mechanism by which PFAS cause their effects, this is still quite unclear. There's a number of biomarkers that you see here that are being changed upon PFAS exposure, and a number of them are also considered to be mechanistically related to the adverse effects that are clearly being caused by PFAS. There's no discussion about that, but the mechanisms are quite under discussion. Specifically, PPR, gamma and alpha are being considered, but also other receptors are being considered critical for the toxicological mechanism of action. But even though really huge numbers of publications are out there, many people are investigating PFAS toxicity, but up to date there is still no single mechanism known there where consensus could be reached that this is responsible for the adverse effects. And most probably there is not one single mechanism, but there's a variety of different mechanisms responsible for the adverse effects. Regarding the health-based guidance value, the EFSA published an opinion in 2020 that I mentioned already previously, that you see here: risk to human health related to the presence of perfluoroalkyl substances in food. There were other opinions before, namely not so long before 2020, there was in 2018 another opinion, and then before that also, but the current health-based guidance value is was published in this scientific opinion from the EFSA. The critical effect in this opinion, as I mentioned already, is immunotoxicity, and specifically it was decreased vaccination response that was observed in the critical studies. The study is an epidemiological study where children were investigated, and it was observed that there's a clear dose response of decreased vaccination response upon increasing PFAS intake. There is a discussion, or was a discussion going on about the adversity of that endpoint, because basically the adverse effect would be an increase in infectious diseases, and so far there is no strong evidence for PFAS really causing this increase. Nevertheless, the decreased vaccination response is considered so closely connected to a putative increase in infectious disease that the decreased vaccination response is considered a relevant endpoint. The derivation of the health-based guidance value is somewhat different from other studies. Usually the reference point taken from the dose response data from the respective studies would be an oral uptake leading to an adverse effect. This is not the same here. Here we do have as the reference point a blood concentration of PFAS in these children where a decreased vaccination response was observed. The BMDL10 was calculated to be 17.5 nanogram per milliliter. So this is, as I said, not an uptake, not an oral uptake, but an internal exposure. So it's the plasma concentration, the blood concentration. From that value, through toxicokinetic modeling, an associated exposure was calculated to be 0.63 nanogram per kilogram body weight per day for 4 compounds: PFHXS, PFOS, PFOA, and PFNA. This does not mean that all the other PFAS do not cause the decreased vaccination response. And in fact, for a number of substances, it is highly likely that they do. But the evidence is only sufficient for these 4 compounds. So they are since then known as the 4 EFSA PFAS compounds, because the health-based guidance value is only derived for these substances. And then from this calculated exposure, there's no TDI, no tolerable daily intake, but a tolerable weekly intake, TWI, was derived in order to emphasize that there is a strong accumulation potential because these substances have such a long half-life. And this TWI is 4.4 nanogram per kilogram body weight per day. Of course, again, only for these 4 compounds. With that, we're already at the end of the second chapter of the hazard characterization. I've just shown you the TWI, the health-based guidance value from the EFSA opinion 2020. As a breakout question here at this point, I'd like you to stop the recording for a minute, the presentation, and check what other EFSA opinions were available before that. They're all available online. And check how the health-based guidance values developed over time. So which other TWI or TDI values were out there from EFSA. And maybe you can also consider, for those of you who know that already, You can also check with which other guidance values are out there from other organizations, be it national risk assessment authorities, be it authorities also from outside Europe. There is a number of reference values, and I'm not referring to legal maximum levels in food or drinking water or such a thing, but really health-based guidance values. And do check what other values are available. The third chapter is about the exposure assessment. So now we deal with the question of how much PFAS do we actually take up into the body. And first of all, I would like to show you how the concentrations in the environment have developed over time. What you see here is temporal trends of PFAS in falcon eggs and in seal liver, so not really representative of food items, but these two sample types are indeed representative for environmental PFAS concentrations. And what you see here is that in the period between the 70s up until 2007, there was in both sample times in almost all substances strong increasing trend that you can see here for the falcon eggs and also here for the seal livers, indicated by the 3+. But then in the last 7 or 8 years of this observation period, there is no increasing trend. In fact, there's either no significant trend or even a decrease. And the same is true here in seal liver. So an important thing to keep in mind is that concentrations in the environment are fairly large. They have been huge in the past, but currently we do see a decreasing trend. And now we look at the numbers that are actually present in several food items, because this is the major source of exposure. Now this table and also on the next slide has very many numbers. You don't need to look at all of them. It's really just to get an impression. First of all, let me show you what you see. You see the different compounds, the 4 EFSA PFAS compounds, P4S, P4A, PFNA and PFHXS. And then you see various food items shown here. On this table we don't see any fish, but we do see fish on the next slide. So as I said, really a lot of numbers, no need to look at all of them, but just to get a clear idea or a rough idea, there is in many cases we do have very little or small PFAS concentrations. You see here percent left sensor data and then lower bound and upper bound. So basically this is what you need to look at. And then what you see is there is indeed quite a huge difference when you look at particular food items, especially here in the non-fish food samples, food items. We have a clear increase or much higher concentration than offals. So this is particularly liver and kidney, but also heart, stomach, and various other offals that are maybe not consumed by the majority of the population, but by a relevant fraction of the population. And you see particularly from game animals, so wild animals, not farmed animals, in comparison to all the other food items, you see a huge concentration increase. This is particularly true for P4S, not so much for the other compounds, but you can see when you look at it, you also have much higher concentrations in comparison to the other food items seen here, also for P4A, also for PFNA. Not so much, at least for the lower bound data, for PFHXS, but also in the upper bound data. So this is something that we can clearly take here from this table, that there is variable concentrations in the different food items, but offals is really with huge distance, the highest concentrations can be found here. And then we have the fish data. And also here we do see quite some differences. And then when you look at carp, eels, and offals again. This is also true for fish, but also fish meat, carp, and eels here have fairly high concentrations in comparison to the other fish species. This again is most pronounced for PFOS, but you can also see that here, for example, for PFOA. What we've previously seen were the concentrations of PFAS in the different food items, but of course what is interesting to us in the exposure assessment is not the concentrations only. This is a very important parameter, but what we actually want to know is the uptake. So the dietary exposure, how much of the PFAS do we take up into our body. And this is what you see here. This is a summary statistic taken from the EFSA opinion. You see the dietary exposure for the different consumer groups, the different age groups, from infants and toddlers up to elderly and very elderly. And then you see various exposure scenarios. So first of all, we have the mean dietary exposure here on top, and then we have the 95th percentile exposure on at the bottom. So these are people consuming mean range of food items containing PFAS, and these are high exposures. We also see stratification into lower bound assessment and upper bound assessment here in both cases, and then we have minimum, median, maximum. And you see many numbers. What you need to compare with is the numbers that you see here, so the exposure, the actual uptake, with the health-based guidance value. And if you remember, the tolerable weekly intake is 4.4 nanogram per kilogram body weight per day. What we see here is actually not the weekly uptake, but we see the daily uptake. So here we have the numbers are shown in nanogram per kilogram body weight per day. If we calculate the TWI into a daily exposure, it's approximately 0.6 nanogram per kilogram body weight per day. So the tolerable daily intake, we only have a tolerable weekly intake, but if we would calculate that to estimate a tolerable daily intake, it would be 0.6 nanogram per kilogram body weight per day. And if you have that number in mind, I think you see immediately, even though we don't need to discuss all the single exposure scenarios, you see that the dietary exposure is very high and basically always exceeds the tolerable level. We in fact only have a few scenarios here for the minimum number, lower bound assessment of mean dietary exposure, where we have less than 0.6 nanogram per kilogram body weight per day. And in all other scenarios, we do have much higher numbers, and they really range very much. They are really very much exceeding the tolerable weekly intake that has been derived before. Now we're already at the end of the third chapter with the exposure assessment, and the breakout question here is, what are other PFAS exposure sources apart from food? We have seen in the tables before that a number of different food items contribute to the overall exposure. Fish is a very important food item, and then we've also discussed offals, which maybe is not consumed by so many people, but for those who consume it, it's really the most important source of exposure. But there is also other exposure sources for the consumer, and for this breakout question, I would like you to stop the presentation for a minute and try to identify other sources where we take up PFAS from into the human organism. And then we're at the last chapter, the risk characterization, where we sum up all the evidence that has been brought up by hazard identification and characterization on the one hand, and the exposure assessment on the other hand. And I would like to mention the BFR opinion again, that I have shown you before. As mentioned before, the BfR is the German Federal Institute of Risk Assessment. BfR publishes opinion on a regular basis on various topics that are of toxicological importance, and of course PFAS is of high importance. After the EFSA opinion of 2020 came out, the BfR published this opinion. You can find it online and then download it for free. And for risk characterization, I always find very useful the risk profile that the BfR now publishes with their opinions. And here you see the BfR risk profile from the opinion just mentioned, specifically referring to the decreased antibody response after vaccination. What you see here, A to E, affected are the general population and specifically children. I think what is mainly mentioned here, or children are mainly mentioned here because the endpoints are referring to children, but of course that also affects the general population and is meaningful for everybody. Then we have B, the probability of the health impairment if the tolerable weekly intake is exceeded. It is possible. It does not mean that it's likely. The TWI is fairly low. It is fairly conservative, so that does not mean that there is immediate severe health impacts if it is exceeded, but it is possible. Of course, that depends a lot on how high this exceedance actually is. So if the tolerable weekly intake is exceeded by a factor of 1.5, of course that's much different from an exceedance by a factor of 100. And then another important thing is how long this exceedance actually takes place. Then we have point C, severity of the health impairment when the tolerable weekly intake is exceeded. We do have moderate impairment here possible because there's a number of infectious diseases that can be related to this decreased vaccination response. And of course that depends on the specific disease, but certainly that can be at least moderate in my point of view. Significance of the available data is considered medium. Some important data are missing or inconsistent. That I would think is mainly reflecting to the fact that there is no data on the infectious disease incidence itself. So basically what we're looking at is a biomarker with the vaccination response decrease. But nevertheless, there is a lot of data on PFAS, and the evidence from the epidemiological studies is also supported by the animal studies. What also is missing here is data from many other compounds. As I mentioned already, we do have the 4 EFSA compounds that are now being considered here for this tolerable weekly intake, but a number of other compounds do not have any toxicological studies so far, have not been characterized properly, and I'll come to that point later on. And then point E, controllability by the consumer is not really controllable, even though I would say in some cases there is controllability through avoidance, particularly when we're talking about the offals that we've mentioned. The population group frequently consuming offals does have the possibility of controlling the PFAS associated risk by reducing the consumption of offals. Another risk mitigation measure is the legal setting of maximum levels. This is being done in this Commission regulation that you see here, which regulates the maximum levels for contaminants in food, not only for PFAS but for many other compounds. But PFAS are also regulated here in this regulation. And here you see the numbers. So again, as for the EFSA TWI, we do have the 4 EFSA compounds, P4S, P4A, PFNA, and PFHXS. And we also have a sum parameter here. So we do have individual legal limits, and we do also have sum limits. And then they are different for the different food items that you can see here. At the end of the risk characterization chapter, I would like to mention relative potency factors. I already mentioned before that we do have good toxicological data for EFSA compounds, but for many other compounds, we do not have sufficient toxicological data, and the question of course comes up how to deal with these data, with these substances. One of the major compounds of how to deal with such a situation is the concept of relative potency factors. And you see here there's a number of publications out there dealing with relative potency factors for PFAS, and you see here one of the latest very important publication on relative potency factors that make suggestions of how that could be put into practice. But in fact, so far these relative potency factors are not being adopted by regulatory authorities and being part of the risk assessment, so that has not been done so far. We don't have time enough to explain all the details of the concept of relative potency factors, but this is for this last chapter the breakout question: what are relative potency factors? So for those of you who are not familiar with this concept, you can look at the publication that I mentioned before in the slide before, or you can also just search on the internet what is available about relative potency factors so that you can get an idea. For those of you who are familiar already with the concept of relative potency factors, you can specifically look at the publication mentioned before, which type of potency factors have been suggested for PFAS. There's also a number of other publications dealing with that topic. You can look at that. And for those of you who are completely aware of all of that, you can try to identify identify why, or answer the question why these relative potency factors have not been put into practice so far. There is some, on national level, some exceptions to that, but the EFSA so far does not or did not adopt the potency factors, and the question is, why is that the case? What other data would be needed, and when would we expect to have these relative potency factors put in place? Okay, and with that we're already at the end of this lecture. So we looked at the 4 different aspects of the classical risk assessment. In the first step, we looked at the hazard identification, and we've seen the chemical characteristics of PFAS compounds, particularly with the very stable carbon-fluorine bond. We looked in the hazard characterization at the various adverse effects, and we've seen that PFAS can cause a variety of different effects, with a strong focus on liver toxicity and immune toxicity. We looked at the exposure assessment, where we really saw many numbers. As I mentioned, there's no need to look at these in detail, but what is important is that food items are a major source of PFAS exposure, and namely fish, and even more so edible offal, is certainly these are the major contributors. And then in the final step, we also looked at the risk characterization and at the risk profile from the BFR, the German Federal Institute of Risk Assessment. And with that, I'd like to thank you for your attention.